All right, so when a host cell is actually infected by a virus, there are some different things that can happen within a cell that can be used as a diagnostic tool for medical professionals. So these damages to host cells that are infected with viruses are called cytoplastic effects. So that if we went and looked at cells that were infected with viruses underneath a microscope, then we could look and see, and sometimes we see multinucleation. Sometimes we see different inclusion bodies, but we can detect something that is wrong with that cell to know if a host is infected with a virus. It's not as easy to detect if people have viral infections as it is if someone has a bacterial infection. A lot of times we can do things like we can take urine samples and count the amount of bacteria that are found in urine to determine if someone has like a UTI. Or you could take a sample of pus or a throat culture, and we're looking for, a specific kind of bacteria that's causing that kind of infection. But with a virus, they're so small, we can't see them microscopically, that we can't grow them in a lab, that it's a difficult thing to diagnose. But this is one way that we can do it, is by observing these cytoplastic effects. Now, don't worry about memorizing cytoplastic effects for different diseases, but I just want you to kind of be aware of some diseases that have these cytoplastic effects. Cells that are infected with a smallpox virus look very round. They have multiple inclusion bodies. Adenoviruses, there's other nuclear inclusions. Herpes have multinucleated situations where they look like they have more than one nuclei. So there are some common viruses, influenza, that we can actually detect how cells are behaving in the presence of those viruses. Now, like I said, it's an important diagnostic tool because we can't really necessarily sample for a virus in a lot of different kinds of situations. Now we're kind of finding out all sorts of information from the media. Being in the middle of this pandemic with coronavirus, I'm sure that you're probably a lot more familiar than my students typically are in years past on how viruses could be tested for or detected based on, just looking for their genetic information in sampling. Another thing to keep in mind about viruses is that viruses can remain in a person's body latent for a long period of time. So viruses can become inactive within a cell, but then can become periodically activated. So if you think about like herpes or even about chickenpox and shingles, you kind of carry that around, and then all of a sudden you can have an outbreak of that. Mono kind of works the same way. Sometimes you can carry mono around or be tested for positive for mono even after you're asymptomatic, and sometimes people have reoccurrences of symptoms. Some other things to think about with viruses is that some viruses can go in and they can alter the genetic makeup of the cell that they are infecting. And this process is called transformation, when a virus transforms or changes the DNA. And sometimes this can lead to an issue with the cells that leads those cells to be cancerous or be tumors. In this case, viruses that cause this situation are called oncogenic viruses or oncoviruses. So they're capable of initiating tumors in humans. Now, where this also becomes relevant or medically significant to us is with bacteriophages. Bacteriophages are actually viruses that infect bacteria. However, how it kind of applies to the last idea is sometimes these viruses can infect bacteria, and they can change genetic information within the bacteria that actually makes those bacteria more pathogenic to humans, right? So again, a virus that is infecting a bacteria but then makes the bacteria more dangerous. I'm going to walk through bacteriophages. We'll talk a little bit about them here. Bacteriophages are mostly double-stranded DNA viruses. They look like this. They look like little aliens, right? And they infect bacteria. They're also a genetic vector that we use in the development of vaccines or in the development of altering bacterial cells or other cells' genetic information. I'll talk maybe a little bit about that later or later in our genetics lecture. All right, so there are these crazy looking alien things where they have these little legs, but they're basically just made up of just proteins. Sorry, I was just teaching my chemistry class, so I'm thinking protons in my head. But proteins. So they have this icosahedral head where the genetic information is stored, and then they have these little legs. I mean, they kind of look like little legs, and they will adhere to the cell wall of the bacterial cell, and then they kind of squat down as they adhere to this, and they will inject their DNA inside the cell. So these ones aren't actually invading and getting inside of the cell, they're injecting their DNA. And this is actually an electron micrograph of that happening. Right, so the little process of viral replication for bacteriophages is a little bit different because of that. So if we have a host bacterial cell here, the bacteriophage comes, it attaches and absorbs to the surface of the bacterial cell. It does its little squat and it injects its DNA in. So the penetration process isn't the entire viron getting into the bacterial cell, but just the genetic information. And then that genetic information is used by the bacterial host cell, and it is replicated to build all of those components that are needed to rebuild the virus. That first stage is called the eclipse phase, and then the second stage we call the viron phase, where all of those little parts assemble back together. So we have that assembly and the maturation where we form this sort of, seemingly like complicated little bacteriophage shape, and then they will actually replicate themselves so many times until they've lysed the cell, and then they are released. All right, so a little bit different than a eukaryotic virus, these bacteriophages infecting bacteria. This is their viral replication process. So here actually shows the steps of that happening. You can see over here on the left-hand side, you've got all of those little vessels for the genetic information, all those little bacteriophages. They have absorbed to the surface of this. They have injected their DNA. And then over here on the right-hand side, you can see where the cell has lysed. After those new virion are formed and are lysed from the cell, they go off and infect other cells that are adjacent, where these guys are pretty much just empty protein shells, right? Now, that viral DNA can also inject itself into the bacterial DNA. So again, this is how they would change the bacterial DNA or transform the bacterial DNA in order to make it so that that bacteria may be more pathogenic if it doesn't necessarily kill it, okay? The other thing is this is how this can be used for purposeful genetic transformation. So we can take DNA or little pieces of DNA that we want, and we can put them into a bacteriophage, and we can actually infect maybe a coli cell with that bacteriophage, and it will inject that piece of DNA into the bacteria that we want to put into its genome, right? So a form of genetic modification. Right, this slide just kind of shows you a side-by-side comparison of the bacteriophage viral replication process and the animal viral replication process if you want to look back and review those two things. All right, the last thing that I'm going to talk about in this video is how do we actually study viruses and how do we treat them? They're very different than antibiotics or than bacteria. We cannot grow them in a lab like we do in our lab in class, right? We can't just put them on a plate and put them into an incubator. They need a host to survive. So when we're growing virus or in the lab, we need to do it in lots of different kinds of ways. There's different options. One, we can put it into a live animal. So again, this isn't ideal. I mean, a lot of people get kind of squeamish about this kind of thing, but it's not really ideal. People don't really want to deal with handling animals in a lab if they don't absolutely have to for medical research. It's expensive, they're messy, there's a lot of federal regulations, so not necessarily the most ideal way of doing things. But we can expose animals to a virus, and that viron will perpetuate inside of them, and we can grow multiple viruses that we can study in those host animals. Now, an easier way to do this is to use bird embryos. So essentially an egg that we inject with the virus, and the cells inside of the egg will get infected with the virus and then grow within there. This is also the reason why some people have allergies Like if you have an egg allergy, there's certain types of vaccines that they tell you that you can't get. So this is a real simple procedure, cheap, easy. You know, you got a little carton of eggs, and we use some sterile techniques to insert the virus into the egg to sort of cultivate it within the lab. Now, another way we can do this is with using tissue lines. So essentially a tissue line is just human cells or animal cells that are grown in a culture on a plate that's very similar to what we do, growing bacteria in the lab, and then we infect the cells that are living on that plate with the virus. Now, viruses are the most common cause of infections that we have, and as we're realizing here, they can lead to a lot of death. They can have a high morbidity and mortality rate. They can lead to long-term disabilities, but we don't really have the types of drugs that we do to treat antibiotics, right? Or to treat bacteria. We have antibiotics which work on living bacteria but are ineffective against viruses. There are certain drugs that are antivirals, but viruses are very simple things, not organisms, but they're difficult to stop because there's not much to them. A lot of our antiviral drugs work by blocking viral replication, especially if there's something specific about, maybe they have that reverse transcriptase or some specific kind of proteins that are needed in order to replicate that. Those are what our antiviral drugs are doing. Interferon is a compound that's produced by the human immune system and can be produced in a lab and used, and that can sometimes be used for preventing viral replication. It actually helps to trigger cells that are infected with viruses to stop replicating. But again, not ideal and not as easy as like giving somebody an antibiotic to treat a bacterial infection. So really the best way to prevent the spread of viruses is through vaccination, right? And I will have a whole nother lecture where we talk about infectious diseases, where I'll spend some time and we'll talk about vaccines and the importance of vaccines and all of the sort of controversy and the science behind a vaccination, how it works. All right.